Last time we covered the division or the classification of the nervous system, neuron, its definition, classification. Today in this lecture we shall be covering structure of a typical neuron, synapses and neuroglia. Structure of a typical neuron comprises of a cell body and nerve cell processes. The nerve cell body is also called soma or perikaryon and contains cytoplasm along with central nucleus and organelles inclusions present within the cytoplasm, and all of these structures enclosed by a plasma membrane. Nerve cell processes, they comprise of dendrites and axon. Let's take a look at the nucleus of the nerve cell body. The nucleus of this nerve cell body, it is large, pale, and central in location, with widely scattered chromatin inside it, containing a single large nucleoli. In females, in the periphery within the nucleus, bar body is also present. And nucleus gets displaced in case of nerve cell injury. The cytoplasm of a nerve cell body has organelles and inclusions. Among the organelles present within the cytoplasm of a nerve cell body, we have nasal substance. Now, what is nasal substance? Nasal substance is characteristic of neurons. It comprises of rosettes of polysomes and rough endoplasmic reticulum that is specialized in the protein synthesis specific to neurons. Nasal substance is found not only in the cytoplasm of the cell body, but also within the dendrites of the cell body. But it is not present in the region of the axon hillock and axon itself. On electron microscopy, this nasal substance appears to be a broad cistern. So a number of such cisternae stacked one on top of the other. On light microscopy, it can be seen basophilic, and it is associated with a very important phenomenon called chromatolysis. Chromatolysis is a phenomenon that takes place as a result of retrograde degeneration of the neurons in the central nervous system. It involves the nasal substance appearing as fine granules dispersed throughout the cytoplasm, mostly concentrating themselves around the periphery of the cell body. So in the times of injury to the cell body of the neuron, nasal substance gets dispersed towards the periphery of the cell body, so that under the microscope it gives an impression that nasal substance has disappeared from the center of the cell body. So this phenomenon, in which due to retrograde degeneration or retrograde injury to the cell body, nasal substance moves towards the periphery of the cell body, giving an impression that it has disappeared from its normal position. This phenomenon is called chromatolysis. Then other organelles, they include the Golgi complex, which is present in the form of wavy threads scattered again throughout the cytoplasm. Then we have mitochondria, the powerhouse energy stores of the cell, lysosomes, we can see containing the hydrolytic enzymes for the debris digestion. And then we have some components from the cytoskeleton, which include the neurofibrils, microfilaments, and microtubules, responsible for the transportation of the different elements throughout the nerve cell body and its process, as well as providing and maintaining a shape to the neuron. Then we have centrioles. The centrioles in the neuron, in the immature neuron, they may be associated with the division of the neuron, but in a mature and fully developed neuron, these centrioles, they reduce their function to the division of the microtubules. Then we have certain inclusions. These inclusions may be found within the cytoplasm of the neuronal cell body as a result of either metabolic byproducts within the cytoplasm or as a result of certain diseases. Action potential across the nerve cell body. It is a much more detailed topic belonging to the physiology. And there you will be taught how the nerve impulse is conducted along the neuronal process with the processes of depolarization and polarization, in which there is influx and outflux of the negative and positive ions across the cell membrane. Then we have neurites. Neurites are the processes or the extensions of the neuronal cell body, and there are at least two different types of processes associated with the neuronal cell body. These are called the dendrites and axon. The dendrites, these are the short processes arising from the cell body, multiple in number, and they taper off in their diameter and branch profusely. In the central nervous system, some of these dendrites, they may bear short projections called dendritic spines. The cytoplasm of the dendrites is almost similar to that of the perikaryon. These dendrites, they function to increase the surface area of the cell body as well as conduct the impulses towards the cell body. Axons, on the other hand, they are the long processes arising from a conical projection on a cell body called axon hillock. Here in this region, you can see there is a conical projection coming out of the cell body forming the axon. This region is called axon hillock. And this region is devoid of nasal substance. The axons, they do not branch near their region, but they rather branch near their termination. And sometimes their ends enlarge to form swellings called terminals. Particularly some neurons in the autonomic nervous system, they may show a series of swellings like beads on a string called varicosities. Just like any other cell, the axon has axolemma, exoplasm, but this exoplasm is devoid of nasal granules and Golgi complex. So these two organelles are absent within the cytoplasm of the axon. So this means that axon is dependent for the proteins on the cell body. The axon functions to conduct the impulse away from the cell body, but in dorsal root ganglion particularly, the function is reversed. It is said that larger the axon, faster is the conduction by the neurons. The initial segment, first 50 to 100 micrometers of an axon, is labeled as initial segment, and it is the most excitable part of the axon. It is where the action is generated, action potential is generated under the normal conditions. So such points can be asked in the MCQs by the University of the Health Sciences. Now let's see some features related with the transport across the axon. The transport across the axon is important for the availability of different types of neurotransmitters, proteins, cytoskeletal elements from the cell body or between the cell body and the axon. So the transport can be of two types: anterograde, that is forward movement, and retrograde, that is backward movement. Forward movement towards the axon and backward movement involves the movement towards the cell body. So what are the characteristics of both of these types, anterograde and retrograde? Anterograde, depending on the speed of the transport, is divided into two types: fast anterograde and slow anterograde. The fast anterograde occurs at a speed of 200 to 400 millimeters per day, whereas slow anterograde has a rate of 1 to 5 millimeter per day. Neurotubules and kinesin, they are used for the fast anterograde transportation, and this method is used to transport the proteins, organelles, and neurotransmitters. Whereas slow anterograde is the method used to transport cytoskeletal elements and protoplasmic elements. Retrograde, however, is the backward movement towards the cell body. It occurs at a rate of 100 to 200 millimeter per day. Takes up the nerve growth factors like neurotropic viruses, and growth factors, as well as some viruses that attack the neurons like herpes zoster, polio, rabies, and tetanus. And retrograde is the type of movement that uses neurotubules and dynein. So kinesin is for the anterograde and dynein is for the retrograde.